This video lecture is over Chapter 4. We're going to be covering extensions to Mendelian genetics, and a large portion of this chapter is understanding the vocabulary and what the vocabulary means and how those deviations from Mendelian genetics could potentially affect you if you were doing a genetics problem. So since Mendel's work was rediscovered in the early 1900s, there's been a lot of researchers that have looked at ways in which the genes influence a... Since Mendel's work was rediscovered in the early 1900s, there's been a lot of researchers that have looked at the ways in which genes influence an individual phenotype. And what we've found is that a lot of these influences are not behaving in a strictly Mendelian manner. And so we've called these investigations neo-Mendelian genetics, meaning new Mendelian. And some of the ratios that we find that don't conform to Mendelian ratios have since been explained. So before we get too far along, let's cover some topics of neo-Mendelian genetics and get our vocabulary straight. So first off, a mutation. Mutation is the source by which we get new alleles in a population. During DNA replication, base pairs get mismatched, and a mutation can occur within a gene. If that mutation causes the protein to function in some new manner, that's ultimately the source of any new allele that comes into a population. Now, just because genetic information gets modified, it doesn't always produce an altered gene product. You could have a mutation that doesn't result in a change of that protein. So we only get new phenotypes when somehow the functional activity of that gene product gets altered. So for instance, if we had an enzyme and a mutation then causes that enzyme to no longer function, that would be a new phenotype. We could change the efficiency by which that enzyme functions. Maybe it functions better or worse than previously. Or in some mutations could actually cause that enzyme to have a completely new function altogether. Because individuals can only carry two alleles at a time, we use the term wild type alleles to talk about what are the most common alleles in a population. They occur the most frequently in nature, and they're usually, but this is important, not always dominant. It's the normal phenotype. And this is the default state for most of the proteins that we say function normally. So we have the right amount made, they function normally. When we have alleles that then altered that wild type allele, so we have a mutation that alters it, we then get what we call mutant alleles. These tend to be, by their very nature, rare in natural populations because most alterations to the protein are going to make them function less well. Therefore, they won't be retained. So, mutant alleles are often inherited in a recessive fashion. So how do we distinguish between these alleles when we're doing genetics problems? Well, dominant, we've already talked about that. It's usually denoted by an uppercase letter. Sometimes it'll be in italics. Recessive is usually in the lowercase in italics. And sometimes we'll see a recessive with, say, with a superscript on it. If there's no dominance, then the uppercase letters and superscripts are used to denote our alternative alleles. So, for instance, if there's no dominance or recessive, we could have allele 1, so the R1, the R2, or CW, CR. As an example, let's look at a gene that that encodes for body color in Drosophila, which is the fruit fly, and we could have what we call the ebony mutant that makes that fly black. The normal would be gray. And we denote this wild type generally with a plus. So wild type is plus, and the mutant is lacking that. And this is generally what I will use a lot when we start getting into some more complicated genetics problems. I'll use the plus denoting wild type, and I'll use the letters to denote the mutants. So what are some things that we have discovered in different traits and different organisms that don't adhere to Mendel's initial postulates? One of those is what we call incomplete or partial dominance. So sometimes what we find when we make a cross is that the offspring don't have the phenotype of the either parent, but it's kind of a mixture of the two. So for example, here we're looking at these primrose flowers, right? And we have a blue, we have a white. The blue is the dominant, the white is the recessive. But when they're combined, we get an offspring that's pink. Okay, this is a blending of phenotypes, and it's not complete dominance; it's incomplete dominance. So the red is still kind of coming through, but not fully. It's incomplete. Another aspect that we have is what we call codominance, in which we have two alleles at a locus that produce different and detectable gene products in the heterozygous. So we don't have dominance or recessiveness, and we're not blending. The best example that I can give you guys for this one is blood type, right? So in blood type, we have a series of immunoglobulins that produce antigens. So we have the A, the B, and then the immunoglobulin without any antigen, the O. And if you're A, O, then A is going to be dominant to O. If you're BO, B is going to be dominant to O, right? So we'd have blood type A, blood type B. But if these individuals then have children, they could have an individual, or they could have a child that's AB or, AB. And AB is codominant. So you produce both the antigen for the A and the antigen for the B. Okay, so both of those antigens are being produced. A is not dominant over B, B is not dominant over A, both get produced, so it's codominance. Additionally, we've discovered that there's a lot of traits that are characterized by a distinct phenotype, and they're affected by more than one gene. It's not a single gene that does this. This gene interaction is the byproduct of numerous gene products that contribute to the overall development of the phenotype. This is true of so many of our phenotypes and traits. Great example, if we look at just coloring in bell peppers, okay, there's two genes involved. We have this R gene and this C gene. Okay, if you're red, you're dominant for both the R and the C. If you're green, you're recessive for both the R and the C. Okay, that gives us our two character states, the red and the green. However, when we start doing a cross between a red and a green bell pepper, what we get is we get orange bell peppers, we get yellow bell peppers, we get green bell peppers, and red bell peppers. And we see that when we're homozygous dominant for red, but homozygous recessive for that green state, for the C, we get an orange color. It's not an intermediate, it's a completely new phenotype. Likewise, the inverse of that too is when we have homozygous dominance for the C, and we're homozygous recessive for the R, we get a yellow phenotype. Okay, so this is a great example of just how we have multiple genes and we get these unexpected phenotypes that are become kind of different to, or difficult to figure out what's going on from a strictly Mendelian fashion. Likewise, if we go back to the blood type, right, 3 alleles, we have the isoagglutinogens and that produce the antigen. So we have the A antigen, the B antigen, and then we have the I allele that produces no antigen, which we denote as the phenotype O, right? And the AB exhibit codominance, right? So we're codominant, but yet the A and the B are dominant to the O. This chapter also brings in the concept of penetrance and expressivity. So when we're talking about penetrance and expressivity, we're talking about the degree of which an expression of a particular trait is found in a population. So penetrance, this is defined as the percentage of individuals that show at least some degree of expression of a mutant, of a non-wild type genotype in a population. And we can really kind of divide it into what we'd say is complete or incomplete. So complete penetrance would mean if you have that allele, you find that. So like Mendelian traits in our pea plants. If you're tall, which was dominant, all peas that had the dominant allele are going to express dominance. So we'd say that trait shows complete penetrance. There's other traits, however, that we find are incomplete. We don't always find them. Somebody could have that allele, but it doesn't necessarily mean that they're going to exhibit the phenotype. A great example for this is the BCRA1 allele, commonly referred to as BRCA, right? This is associated with breast cancer. It's in women or in men that have the BCRA1 allele, they tend to develop breast cancer. But it's not if you have it, you're going to get breast cancer, because what we find is that only 70% of the women carrying that allele develop breast cancer by the age of 70. So there is an increased risk of disease by having this allele, but it's not 100% penetrant. It's incomplete. There's an unknown certainty of whether you're going to get that disease or not. Now, expressivity is similar to penetrance, but now we're talking about the range or degree of expression of the phenotype may differ from one individual to another individual. So can we have some differing expressivity in these traits? So you could have a dominant allele that's not expressed in a heterozyte individual. For instance, polydactyly. This is where you have more than 10 fingers or toes. It can be, it's a dominant allele, yet not everybody who has polydactyly is going to have a complete extra digit. You could have something where you just have a little bit of it. Take a look at this. So example, right here, see, here's this individual where they have this small extra digit, but here's somebody that has a complete extra digit that's come in here, right? So the polydactyly shows different expressivity. So just to recap, penetrance is the percentage of individuals that show at least some degree of that expression. So if you show this, that would be penetrant. That's also penetrant. But expressivity in this would be very different, right? This is higher expressivity than here. And obviously here, really high expressivity. Going along with this in expressivity, we sometimes see genes that then get affected by other like environmental factors. We can have temperature effects. So this is seen in both primrose that has those red flowers. It'll get a red flower if it blooms at 73 degrees Fahrenheit or warmer. It's white flowers if it's down at like 64 degrees. Okay. Siamese cats or Himalayan rabbits. Okay, again, here's some difference in expressivity of the same gene that's giving coat color. There's a temperature effect. So in areas in which it's cooler, so like on the ears where we have a lot of evaporative cooling, nose, on the legs, here the enzyme loses its function, and we then get this darker fur coloring in the rabbits and in the cats than in the rest of the body, where that enzyme still has its function. We can have nutritional mutations in which you may lose or lose the ability to synthesize nutrient molecules due to things in the environment like age. So lactose intolerant, like over time, right, you lose the ability to produce the enzyme lactase, and suddenly you can't you can't digest lactose, but we're able to digest it early on. So the expressivity is changing over time. Other things that deviate from from Mendelian genetics is when we have multiple alleles. So not all systems are just two alleles. There's plenty of traits in which we have a multiple allele system. And even though an individual can only have two alleles for a single gene, there can be multiple alleles in the population that causes lots of different phenotypes. Now, when we look at multiple alleles, we should note we study this in populations. We don't study this in individuals because an individual can only have two alleles. But in a population, there can be multiple alleles. If we look at coat color in a lot of different animals, here we're looking at rabbits, we can see we have multiple alleles. We have what is the wild type, which would be brown. There's the chinchilla allele, okay, that is showing incomplete dominance over brown and Himalayan. We have the Himalayan that we talked about. This is another allele, right? It causes that temperature effect. And we can also have the mutant albino, in which we get a solid white, right? Only occurs when we're homozygous C. So 4 different alleles, and depending on the combinations that you can have a lot of different phenotypes. Something else that's going to deviate or cause our Mendelian expected values to change is a lethal allele. So if we have a lethal allele, this has the potential to cause death in an organism. And if it's an essential gene, it's absolutely required for survival. It's going to lead to what we call a lethal phenotype, and it's never going to be able to survive or reproduce. It's estimated that about a third of all of your genes are essential for survival, and so they do not tolerate mutations very well. Mutations tend to lead to lethality. If we do have a lethal allele, and it's inherited, usually it'll be inherited in a recessive manner, and time of death can be dependent upon when the gene product is essential for development. If it's essential early on, then you're going to die earlier. If it's not essential early on, then maybe it's not going to be, it'll be a late onset. If you're heterozygous, you may be able to tolerate a non-functional mutant allele, but it may make it harder for you to survive. You can tolerate it, but maybe it's harder for you to survive. Why are these important? Well, lethal alleles can change our ratios really quickly. So if we look at it in mice, in their coat color, there's a yellow coat called a gouty, and a gouty is the normal, the yellow is the mutant. So here's an agouti mouse. It's normal, but if we have the yellow allele in a heterozygous state, it'll be tolerated. But if we're homozygous for the yellow, so for AY, we die, and it will not be born. What that means is instead of having a 3-to-1 ratio, we actually, in this gene, have a 2-to-1 ratio. So for every 3 mice born, 2 will be agouti and 1 will be yellow. We will never have that 4th mouse being born because they will all die. This is important because when you do Mendelian genetic problems and we talk about a lethal rate, you need to change your ratios. I believe we did that last time in the previous PowerPoint when we went over the Tay-Sachs disease. How do alleles become lethal? Well, these mutations usually tend to prevent cell division, and that causes an organism to die at an early age. There are a few that are later onset, like Huntington's disease is a good example. Tay-Sachs disease is a good example. We mean later onset in the sense that in Tay-Sachs disease, maybe you can be born normal, but after a few months it starts to be diagnosed. You're not able, you lose, or you don't have the ability to break down the gangliosides that exist in our nervous tissue because we lack the enzyme, the hexoaminidase, which helps break us down. And so generally with Tay-Sachs disease, children are going to die fairly early. Now, another deviation from Mendelian genetics would be what we call epistatic genes. Epistatic genes are genes that hide, they mask the effects of another gene's alleles. And we can break these into what we call dominant epistatic genes and recessive epistatic genes. In a dominant epistatic gene, if we are homozygous for the mutant allele at one or two more of those genes, we're going to be able to produce the phenotype, and it'll be the dominant phenotype that we see. Recessive would be the opposite. Let's look at. Let me give you an example. This will make a little bit more sense. Hair color in horses. Okay, 3 genes play a role into what type of color we find on the coat of a horse. We have 1 gene that we designate W. That gives us the presence or absence of color. If we are homozygous dominant for W, it's actually a lethal allele. So there's no epistasis. The effect of that will be death. However, if we're heterozygous, so we just have 1 copy of the W and we're not able to produce color, what we'll get is an albino horse. Okay, it doesn't matter what we are at any of the other genes, we will be albino. So the epistasis of the W here is dominant. It's a dominant epistatic gene. Doesn't matter what we are for everything else, we will be albino because we don't have the ability to produce color. Now, if we're recessive for W, now the other 2 genes come into play, and the interaction between these are going to determine whether we're red or black. So if we have a homozygous recessive for the A, we will be black. Okay? As long as we have a single dominant copy of the E. So we say that the A is recessive epistatic. It blocks the red coloring. If we have a dominant allele for both the E and the A, then we'll be what we call either brown or bay. So the bay is where we kind of have these black hawks and tail. Brown would be, completely brown. But this is what we get. The black isn't expressed and the red isn't expressed. If we're homozygous recessive for E, and it doesn't matter at all what we are then for the A, we could be, even if we're homozygous recessive for A, we're still going to be red. This one kind of trumps the other, but it still is a recessive epistatic gene, and we'll block all black, and we'll have this totally red horse. Red, the brown hair, the reddish coat. Okay, so here we have lots of different epistatics. We have dominant epistatic in the W, and then we have 2 different recessive epistatics in the black and the red. Next term that we want to talk about is pleiotropy. Pleiotropy occurs when we have just a single gene. Okay, so we're not having multiple genes. There's just a single gene, but we have multiple phenotypic effects. So it's not like we have a fixed character state, like you're either tall or dwarf. Instead, that one gene can cause lots of different things. A great example of this is PKU, where we have phenylketonuria disease. Here we have a single gene defect. It's autosomal recessive, and when we have this defect, we're not able to break down phenylalanine. That's an amino acid. It's found in a lot of foods, especially high protein foods. And if we start accumulating this phenylalanine, it can have a lot of effects. Some of those can include things like mental retardation. It can lighten your hair color. You could have skin problems. You could have seizures. The point being is it's different for every single person. It's not like if you have this, then it only affects skin color, right? No, it affects lots of different things. That's a pleiotropic allele. Another great example, eye color. 1 gene, right? Two states. You're either A or G, right? If we're homozygous G, then we have blue eyes. If you have that A allele, though, it's going to be dominant, and you're going to start getting brown. However, there's a pleiotropic effect in that brown. You could have solid brown eye, but a heterozygous individual could have a blue eye with some brown flecks, or it could have what we call this green eye, in which you have the hazel, the brown, and the blue kind of intermix, and it kind of gives that appearance of having green eyes. The exact same genotypes for all 3 of these: brown with blue with brown spots, hazel, and brown. But because it's got that different pleiotropic effect, we aren't able to distinguish it. It's different between people. So make sure you go back and you review the different things that pull things out of Mendelian genetics. These are the deviations. Not everything functioned as simple as how Mendel found it. Things can be a lot more complex. So review this vocabulary, understand this vocabulary, understand what laws it might violate. Is it the law of independent assortment? Is it just dominant recessive? What's being violated in these? This is where your questions will come from.